Welcome to Measurement, Design, and Statistics 1, Week 2. Last week, we ended with the conclusion that good research design rules out plausible rival explanation and increases confidence in the conclusions that we draw. But how can we be firm in the conclusions we draw? The glib answer is good research design, but what does that mean? This week, we'll consider three elements. First, good results are repeatable, not one-time good luck. So we'll consider the importance of replication, which is getting increasing attention in our discipline. Second, we'll consider whether the study is designed parsimoniously to rule out alternative explanation. This is so-called internal validity. Third, we'll consider whether our results, no matter how well designed, actually generalize to other people, settings, or times. This is so-called external validity. Last week, we talked about the hypothetico-deductive cycle, emphasizing inductive versus deductive starting points. But we sort of glossed over the bigger picture here. This is a cycle. You test, evaluate, repeat. Test, evaluate, repeat. But what does it mean to repeat? In its simplest form, it means I do the study and verify I get the same results, or that I revise and refine my hypotheses and methods, but I keep trying to make sure that the core findings are solid. The idea of repeat is bundled under the rubric of replication. This is Al Kasdan of Johns Hopkins, whose book we talked a bit from last week. And in his book on clinical psychology research methods, Kasdan said, tentativeness and ambiguity of research findings can never be completely eliminated. At the same time, the accumulation of multiple investigations can increase the clarity of the findings. That is the impetus for replication. You may have heard of the replication crisis. Although psychology gets picked on a lot because a lot of classic findings, especially in social psychology, have not been replicable, all of science is grappling with the fact that a lot of findings have not been adequately replicated. We're going to spend some time on this topic because, as you'll see, our disciplines and funding agencies are realizing we failed to emphasize replication. In thinking about it, it's a fascinating microcosm of some of the factors that actually govern how science gets done. Some of the screen captures that follow come from this excellent blog by Diener and Biwas-Diener. Did I just see a pirate? That seems unlikely. But if I see a pirate more than once, I'm going to start to believe it's real. That is the point of replication in a nutshell. Major social psychology findings have failed to replicate, but, as the Dieners point out, psychology is not alone in this crisis. In the late 1980s, physicists claimed they'd found cold fusion, but nobody could replicate their findings. Many similar problems have emerged in biomedical studies. In one survey of major replications in psychology, reported by the Open Science Collaboration and Brian Nosek in 2015, only about one-third of the psychological studies in premier journals could be replicated. Yikes. There are two big reasons that we replicate. One is to assess the reliability of our findings, or so-called internal consistency. Are our findings consistent across studies, or are our findings investigator or method dependent? If a training program only works when a particularly effective teacher implements it, heaven help us if less effective teachers are used. It gets tricky. As you know, often only successful studies with statistically significant results, a dubious criterion of success to which we'll return in future weeks, get published. So the problem is, sometimes 20 studies might have gotten dumped into the file drawer of failed studies before one gets published. This is called publication bias. But if published studies are rare strokes of luck, and we never find out about the other ones, then no wonder it's hard to replicate lucky strikes. There are many kinds of replication. A direct replication is a carbon copy, do everything the same. Those are really hard to publish. Systematic replications are those in which you basically do the same study, But you switch it up. Different investigators, implementers, different kinds of participants. Like, does my reading improvement program in Canada work as well in Britain? Some studies build systematic replications right into their initial design. These are the so-called multi-site trials, which are common in phase 3 and 4 clinical trials. At the core is that a study has a central coordinating center, shown here in the large green circle, and then there are multiple sites, here the blue sites 1 through 4. The coordinating center makes sure each site follows standard operating procedures, SOPs, and does frequent quality control to make sure they're sticking to protocol. Now, the study conclusions are not based on the lucky strike of a single site and mix of investigators and staff, but on, hopefully, multiple very different places and people. This is a study I've been doing since 1996, which was funded for a 20-year follow-up in 2017. On the left is a complex 4-arm intervention trial for older adults we did, initially for 10 years. And on the right, you see it was multi-site, coordinated by the New England Research Institutes, but with investigators and participants from very different places. Starting at the top, University of Alabama, Birmingham, Harvard's Hebrew Senior Life, Johns Hopkins, Indiana University School of Medicine, Wayne State University in Detroit, where I was PI, and the Pennsylvania State University. By pooling our data, we managed a sample of almost 3,000 adults aged 65 and older, and our findings were not biased by any single site. You'll occasionally see the term replication with extension. Maybe, for example, an initial trial compared a placebo to a particular high dose of drug, and in the extension, maybe they added a third low dose level. You switch something up, both to expand what you learn and by introducing innovation to make the study publishable. Clinical replications refer to field testing, bringing interventions to the clinical setting. This is commonly known as a phase 4 trial, where many clinics implement an intervention. Conceptual replications are a bit less direct. Let's say I show that taking a daily vitamin E supplement improves memory, and I measured memory with a list recall task, say the Hopkins Verbal Learning Test, where you hear a list of words and try to recall. Now, investigators want to replicate this, but also test the limits of the finding. Maybe another group of investigators uses a nonverbal memory task, say the brief visual spatial memory test, which is memory for images. And maybe a third group of investigators uses not a supplement pill, but provides a diet rich in vitamin E, and they test memory via hippocampal activation in an fMRI. These all test the same hypotheses, vitamin E helps memory, but a variety of ways that tests the idea from many angles. Because of the replication crisis, there are growing resources for replication studies. This is a partial list of places and practices that have made a commitment to documenting and archiving replication, including successful and failed replication. Indeed, the American Psychological Society has implemented registered replication reports. In a nutshell, investigators submit their protocol to the journal prior to collecting data. They get their peer review and reviewer suggestions prior to trying to replicate a study. Usually, these are planned multi-site replication. And here is a really cool thing. If the protocol is accepted for publication, the subsequent results are essentially guaranteed publication, whether the replication is successful or unsuccessful. We'll return to why that guaranteed publication thing might be a real boon for science.